Good morning and welcome to the Corona Biosimilar Symposium. I am really excited about this day, and we put a lot of time and effort in thinking about a variety of speakers who could not just download what we all know about biosimilars. I think there may be, surprise, surprise, speakers with a difference of opinion. And I think that will reflect societal views of biosimilars. We, Corona, thought that the timing of this kind of symposium was perfect. We've recently learned that there will be a biosimilar on the market very soon at risk. We know that biosimilars are all around Europe, and we know that there's some frustration amongst physicians and our patients that they cannot access all the drugs they need to access. My bias, frank bias, is that biologics do good things for my patients. I'm old enough to remember the days of golden penicillamine, where everyone in the clinic looked just horrible and I felt just horrible treating them, although they were fascinating. And actually called literally in the early 80s, because I heard about this drug methotrexate, and said, can we do better? And they invited me down there, a young nobody, and I met another young nobody named Michael Weinblatt. I'm still an older nobody, by the way, but didn't mean to imply anything else. And so we started with methotrexate, and God, that was a miracle. Then we got to all the biologics, and oh my God, did that change the kinds of patients who came into our office. And then we have societal vectors which challenged us to prescribe these drugs to all the patients who we believe might benefit from them. Thus enter biosimilars. And biosimilars, we will hear from a very distinguished speaker panel, are, I think, going to disrupt the marketplace. We have science is advancing, but everyone cannot pay. So we have tectonic plates as science advances. Patients need to access these drugs, and they're expensive. And not every insurance company will pay, and they're tiered. So one might ask, why is Corona even interested in this type of thing? Here we have a fancy Corona slide. So Corona is the only US-based registry that does indeed collect deep data, RCT quality data. Everything you find in an RCT, you find in Corona. From physicians and patients on all drugs in the US marketplace, collected at the time of a clinical encounter. Not only that, we start with RA, which was my passion early on, and then we have evolved. We have a psoriatic arthritis registry. We have a spondyloarthritis registry. Last year, we started a psoriasis registry, and we will be early next year starting an inflammatory bowel disease registry and an MS registry. We have 15 years of experience. We, primarily moi, has made every mistake in the world. And I've yet to make some. But like every company that's been in existence for 15 years, in order to maintain that existence, you have to learn from your mistakes. You have to learn how to do it right. So we think that experience will position us well for all these other registries because we have the experience. So this is not just about Corona, but the reason I'm giving you this background is to give you context. And the context will hopefully establish for all of us why Corona is well positioned to this fascinating new area which will be disruptive of biologicals. Corona provides real-world evidence. We can do comparative effectiveness. And in fact, we have published many articles on this through one of our superb rheumatologist epidemiologists, Leslie Harold, in the back. Cost effectiveness. Okay, the biosim costs less, so what? Is it as good? Is it better than, say, triple therapy? Is it better than conventional DMARDs? So I think comparative effectiveness without cost effectiveness is half the story. We can provide all toxicities, and all toxicities are followed up at the office with uniform data collection and QC. They're all defined in the same manner across all of the indications. So we have internal credibility that we don't call the toxicities in IBD. We don't treat them differently. We collect the data in the same manner that we do in RA and our other disease states. And so here's where we are. And this is we're in our 16th year. So this didn't happen overnight. So everyone's interested in EMR data. And I think EMR data is everyone's using an EMR now, as opposed to paper charts, which is the way we all started. So here we have this wonderful source of electronic data. So let's, for a second, maybe 30 seconds, look at what an EMR can offer versus the way we're doing it, okay? We're collecting data at the time of a routine clinical encounter in an office setting. All data can contain disease-specific metrics. The metrics in PSA and SPA are different than RA, and of course are different than IBD or IMS or MS. All data identify toxicities in a uniform manner, as I said, across all disease indications, and follow up with confirmation, validation, and adjudication. So if we get a report of a stent, a stroke, peripheral vascular disease, serious infection, zoster, whatever, that triggers another form back to the site. We confirm the event. We get de-identified primary data, and then the event is adjudicated. So we can assign a definite, probable, unlikely category to these data that we collect. These kinds of things simply are not done in an EHR. And that's not to say that EHRs are evil or vile or unnecessary. Of course they're necessary. But I think it's important to distinguish the differences between the way we do things and the way an EHR can do it. So how do we know that our docs are giving us good data? We have the carrot and we have the stick. We realized early on, and I tried to do this, and I was talking to some very well-known data collectors at the time, including Fred Wolf and including Ted Pingus. And they said, Joel, and I don't want to imitate Ted. We all know Ted. I couldn't do a good Ted. You're only going to find so many people who will do this, and it's not going to work. So we realized early on there's got to be something in it for the doc. So we pay him. We pay the doc. In order to do that, we had to have pharma backing. Okay, the stick is you don't collect the critical fields you don't get paid. And we can send you the form back. You missed how long the patient's had disease. You missed a particular comorbidity. Let's say you said pneumonia, but you didn't give us the validation. So we have the carrot and we have the stick. And we have an extensive quality collection of people who drive themselves crazy. And that's the coronavirus. We all drive ourselves crazy because we're so invested in doing it right. We have a sense of internal responsibility as the only entity really in this country who can do this. All right, well, we're almost getting to biosimilars before I stop with this isn't working really great. There we go. So biosimilars will promote competition and innovation in the marketplace. They have to. As we will hear from our distinguished speakers, biosimilars are likely to disrupt, in the true sense of how disrupt is being used in the last few years, the U.S. biologic marketplace. Given the limited studies of the clinical performance of biosimilars, we believe that it is critical that large-scale, uniform collection of utilization, comparative effectiveness, CER, cost effectiveness, and toxicities be collected in the real world as they become widely disseminated. What do we know about these drugs if these data are not collected? What about anti-drug antibodies, also called ADAs? Do ADAs affect efficacy? What about anti-drug antibodies for switchers? Are the clinical effects of ADAs the same across all medical indications? As we know, biosimilars are studied primarily in rheumatoid arthritis and psoriasis, but there's an extrapolation to IBD. So will ADAs affect all the indications for biosimilars in a similar manner? The answer is, we don't know for sure. We should find out. Will that be the case for switchers between different biosimilars? And it's clear that by 1819, there may be patients who switch from the originator to biosimilar A, B, D, et cetera. How is this all going to play out? Who is going to be collecting data? And of course, are these profiles different from the originator or between biosimilars? Forgive me, but I thought that this would be some interesting insights from our friend who died 400 years ago, this year, I believe. What's in a name? That which we call a rose by any other word would smell as sweet. We're all familiar with that. So Romeo, would he not, Romeo called, retain that dear perfection which he owes without the title? Our bloods of color, weight, and heat poured all together would quite confound distinction. Good alone is good without a name. From All's Well That Ends Well. So does Billy Shakespeare get it? Can we boldly apply these quotes to biosimilars, although we can certainly see resonances in what is stated? The U.S. market must address the societal value of biosimilar adoption. There is clearly forces driving the uptake of biosimilars. And I think the forces are absolutely legitimate. Our patients pay too much for drugs. And frankly, the pharmaceutical company doesn't always get what the cost is. There's a middleman, and there's all kinds of backroom deals cut. And I'm dying for 60 minutes to get a hold of that. So biosimilars really have never been widely disseminated in the US as opposed to Europe. This is about to change. Physicians, payers, patients will want to know about the relative real-world value while being reassured about the safety and effectiveness of these newly approved drugs. Patients and physicians will care more about changing a medication, especially if it's mandated, than the molecular data presented at an FDR excellent session, one of which I attended in Silver Spring. They're going to be afraid. So here's our agenda, and I want to have the privilege of introducing our Chief Executive Officer, Ray Hill, before we get to the agenda. I think we have accumulated a wonderful list of speakers from academia, originator pharma, biosimilar pharma, patient advocacy from patient groups, and of course the FDA. After the closing of the individual speakers' sessions, Corona will host a panel discussion, and I am looking for interesting dialogue in the panel discussion. And I very much encourage all of you to participate in the panel discussion. And there will be Q&A after each speaker for a limited period of time. The goal is to consider all of the issues associated with biosimilar use in the US, identifying what is needed and determining how we, Corona, can illuminate the real-world performance and impact of biosimilars and originators in different states is going to be critical. And we frankly think we're the only organization in the US that is positioned to do this right. And finally, this could be working better, sorry. There we go. Merchant of Venice. Truth will come to light. In the end, truth will out. Wherever the data evolve to, whatever the data shows, we do not have a horse in this race. The data must speak for itself. There's limited data, as we know, associated with the utilization of biosimilars. 1 or 2 phase 3 trials, some of which are not even done in the diseases where the drug will be used. We need a good, deep system, an honest system that collects data in the right way to inform all of our constituents, society, yes, payers, yes, docs, most importantly, patients, about the safety and real-world effectiveness of these drugs.